Okay. Yes, so the original talk was something like vision during unconsciousness. I am going to talk a bit about vision during unconsciousness, but I thought I would broaden the scope a bit and talk about generally what my lab does. I know a lot of people have said as kind of a disclaimer that they don't really study consciousness. And I really, really don't study consciousness. Almost to a...
You don't study more consciousness than these guys.
I'm barely conscious. I mean, I'm here, but what we study is vision and what happens when you see. And of course, that's a very big part of consciousness. But we're more on the end of asking how is it that the brain of the monkey in our lab or the human can extract important quantities from the visual input that it gets from the retina. And we ask this in a very mathematical way. What we'd like to come up with would be an algorithm that captures the way in which the system transforms a retinal input into a spiking output that contains information about something like the velocity of a moving stimulus or the identity of an object that you might be looking at. And of course, this question is intimately involved with consciousness. It's probably something that's necessary for consciousness, but it's not sufficient for one to have something like a feeling about what one is seeing or even an emotion. It's more about extracting information in an algorithmic way from an ensemble of signals. So with that, I'll dive into a variation of a diagram that we've probably seen several times now. This is the monkey visual cortex, or at least part of it. And as other people have said in various ways, there are many, many areas that are dedicated to vision in the monkey brain. The largest and probably most important is the primary visual cortex area V1. And from V1, we can trace at least two pathways that are kind of broadly termed a temporal pathway and a parietal pathway, which are involved, again, broadly speaking, in the recognition of objects for the former and things to do with motion and spatial orientation for the latter. So this gives you a bit of a feeling for the anatomy. If you were to take this out and lay it as a circuit diagram, there are many possible circuit diagrams. You've seen several of them. This was one that I found on the McGill webpage, and I kind of like it. They all convey the same point, and that is that coming out of the retina, you have many, many different areas that involve different lobes of the brain, the parietal, the temporal, the occipital. The things to notice here are that you have lots and lots of areas that all essentially depend almost entirely on V1 to learn anything about what's going on in the world. And these are the extrastriate areas of the primate brain. So the first question that one might ask is, given that these areas out here can't know anything about the visual world that doesn't first come through V1, what do you need all these areas for? In other words, they're just reanalyzing information that must first pass through V1 and indeed through the LGN and the retina as well. So you have at least, at last count, 25, 30 different areas just dedicated to processing the information that passes through V1. As you've heard from Julio Martinez and from Eric Cook and from others, probably the best studied example of a transformation from the striate to the extrastriate cortex is from V1 to MT. MT, as you now know very well, is an area that's involved in processing motion signals. It's intimately involved with the animal's behavior with respect to motion, its perception of motion, its control of eye movements, limb movements, etc. So in MT, you find neurons that are very selective for motion direction, but of course you also find neurons in V1 that are very selective for motion direction. And in fact, it's almost certainly the case that the neurons that project from V1 to MT are themselves direction selective. So one might ask, why do you need MT if you already have direction selectivity in V1? And I'll talk now about some work that I did over 10 years ago when I was a postdoc with Rick Bourne. And starting from the very simple question of what do you see when you see this thing moving, the answer, hopefully for all of you, is that you see a kind of a diamond moving purely horizontally left and right across the screen. So this is an important thing to be able to see, obviously. If it were a predator or a prey or something like that, we need to know exactly which direction it's moving in. And we can ask what would happen if we were a V1 neuron. The thing that everyone recognizes about V1 neurons is that they have very, very small receptive fields. One neuron might be able to see a view of the world that is essentially the size of a dime held at arm's length, so about 1 degree of visual angle, or in some cases much, much less. So if you had this tiny, tiny view of the world and you were a direction-selective V1 neuron and your job was to report to the rest of the brain, areas MT and everything else, which direction is this diamond moving in, you can see immediately that you're going to get the wrong answer. Now, because of this impoverished view, you've come to the conclusion that this edge is moving with a vertical component. It actually looks like it's going up and to the right, rather than purely horizontal, which is, as you already know, the answer. This is entirely a property of the geometry of the configuration. It has to do with what's measurable within a very, very small aperture and the orientation of the edge itself. It has nothing per se to do with the brain, but it's an actual fundamental limitation on V1 and its ability to signal motion. So if we go back to our diagram here, this problem that I just described has been known about for almost a century. And other people have studied it in the brain, but we had a different take on it, and we wanted to record from MT neurons in an awake monkey who was just sort of looking at the screen. So this is what Julio would have called zombie mode, I guess. There's no behavior here. He's just sort of looking at the screen. And we're placing a stimulus like the one I just showed you in the receptive field of each neuron. And the question is, simply, does the neuron encode what a V1 neuron might see, which is the wrong answer, which involves a vertical component to the motion, or does it get the right answer? So the reason we think it might get the right answer is that each MT neuron has a much, much bigger receptive field. It can see lots of stuff on the screen, and it gets that by integrating the outputs of lots and lots of V1 neurons that are tiled across space. So when you do this experiment, you get a very clear answer. This is a quick summary of a population response, but on the x-axis here I have time, time from when the stimulus starts moving. And on the y-axis, I essentially have a measure of the error of the MT population response. How much does it actually encode the correct direction, with the correct direction being 0, purely horizontal. And you see the very early responses are way off. They're off by about 30 degrees, and that they're off in a direction that's exactly predictable from the orientation of the edge of the stimulus and from the geometry of a V1 neuron that would provide input to MT. So you see this very early error, which is eventually corrected within about 100 milliseconds, and the corrected response persists for the duration of the stimulus. Once it gets the right answer, it maintains it. If we look at a different edge of the stimulus, so that the edge is moving in the same direction horizontally, but simply oriented in a different way, you get a very nice kind of symmetrical answer where the initial response is wrong, but now it's pointing in a different direction, and it eventually corrects itself. And you can see there's a small residual error in both cases of about 5 degrees. Okay? So this supports the idea that MT, at least if you're willing to wait a few tens of milliseconds, is the area that gives you the right answer, that tells you within a few degrees which direction something is moving in, and motion being kind of a primal aspect of vision. This is an important thing to have in your brain. Then we did a sort of strange experiment. We took the same animals, recorded from the same neurons using the same stimuli, or at least I should say the same populations of neurons, using the same stimuli. And we can plot out the same answer, and it's very different. You again have an early response which is wrong by 20 or so degrees, and it never gets corrected for as long as the stimulus is on. You can do that for the opposite direction too. So this is certainly some kind of manipulation of consciousness. It's anesthesia, so you're also manipulating lots of other things that you don't really understand very well. But it gives you the idea that there's something about the way that these neurons integrate, in this case, motion signals, that changes when you introduce unconsciousness. And I should say that these results do indicate a very specific kind of change in the way the system is working. So it's not the case that the neurons just stopped firing, because what we're measuring here is a measure of direction selectivity. So neurons are very direction selective. In fact, in many cases, more direction selective than when the animals were awake. It's just that they're signaling the wrong direction. They're signaling the very local direction that you get by measuring just one edge of the stimulus. And as I say, this persists for the duration of the stimulus, so it's not a transient event. So for the rest of the talk, I'm going to talk about how is it that the brain integrates different types of signals. How do these extrastriate areas integrate signals that they get from V1 and from lower level areas to produce estimates of things that are important for survival and for vision. And I'm not going to just talk about area MT, talk about lots of extrastriate areas, because it is generally the case that what goes on in MT is also something that should be happening in other extrastriate areas. There are lots of areas that essentially have the job of integrating output that's ultimately from V1 and from the retina. If you look down here in the temporal stream in area IT or TEO, you find cells that respond very specifically to faces. This is one that responds only to a picture of O.J. Simpson. And this is my favorite example of all time. It's probably not illustrative of most IT cells. Most IT cells are not that specific, and they're certainly not tuned for OJ, one would hope. But they have a way of integrating their inputs so that they can provide estimates, they can provide information about what's out there in a way that simply wouldn't happen in V1. So in V1, you might find that cells were very interested in, say, a vertical orientation as it occurs here, but you'd find a vertical orientation here or here, and any one of these types of stimuli could presumably drive the cell. Likewise, you could find eyes or noses or specific shades of gray. It's something about a high-level combination of features that's really necessary to trigger the output of this neuron. And if we could figure out what the relationship was between the inputs that we provide to the animal and the outputs that we measure, we would understand how the brain computes things like face selectivity or motion selectivity. Another area that is of great interest, especially from a theoretical point of view, is area MST. MST gets its input from MT, which gets its input from V1. And this is very much sort of like the O.J. Simpson cell of the dorsal visual pathway. It's not nearly as glamorous in the sense that it doesn't pop out at you as being selected for something very interesting. But it is quite interesting, and it's something that I'm going to spend a lot of time talking about in the talk. The selectivity here is for motion patterns, but we're not talking anymore about motion just moving in a straight line. We're talking about very complex patterns where you see things rotating around in circles, looming towards you so that they're expanding, combinations of those two, so you see spirals of things that are moving. You can see very, very complex patterns. And again, you have the property, which I think is the most important property, which is that this neuron, the single neuron in MST, won't respond to any of the components, any of the individual components of the stimulus, which is its preferred stimulus, which is clockwise rotation of all the elements on the screen in a circle. It won't respond to counterclockwise rotation, and it won't respond to any of the elements of rotation, i.e. motion in a straight line or even pairs of motions in a straight line. So this is not behavior that you would find in MT. It suddenly appears in MST. You certainly wouldn't find it in V1. So the questions I'm going to ask in this talk, there are 3 questions. One is, can we determine the mechanisms by which MST neurons integrate their inputs to produce their outputs? Why MST? Well, MST is a very challenging thing to understand. It's many, many synapses away from the retina. It's at the very highest level of the dorsal visual hierarchy. We don't have good mathematical models of it. Until we have a good mathematical model of MST, we can't really say with any confidence that we understand how these sort of motion responses are computed. And hence, we can't really say what the area is doing. So I'm going to talk quite a bit about area MST. And I'm going to, after that, ask if we can get a mathematical model of MST, does it generalize to the rest of the extrastriate cortex? And this bears on what I think is a really important question in studying the visual system and in sensory systems in general, and that is every little one of these areas in the extrastriate cortex a sort of different entity? Is the brain just a bag of tricks where it solves lots and lots of different problems in completely different ways? Or are there core principles we can extract that apply to each area, and so that once we've gotten a really good understanding of one area, we understand all of them? Obviously, that's what we would like to be the case. And then the third question I'm going to address is, are these computational concepts the keys to understanding consciousness? So to foreshadow a bit, the answer is yes, and no. And or at least I really doubt it. But I encourage people to hang in there anyway because, as I say, I'm not a consciousness person, so maybe people will find a way to sort of put all this together, and that would be an interesting point for discussion. And I will return at the very, very end of the talk to how this could conceivably relate to consciousness, but I'm going to spend most of my time on question 1 and most of the rest on question 2. Okay? So, as I mentioned in the first few slides, there are lots of different kinds of motion that one could be interested in. And motion, as I've said several times, is sort of one of the most important interfaces that you have with the world. You know where things are going, when they're approaching you, when they're running away, and so forth. And we can divide these on essentially mathematical grounds into translation, which is just motion in a straight line, which is what I talked about in the context of MT. Rotation, which is obviously rotation. Expansion, which is probably the most primal stimulus you can imagine. Something that's moving towards you and getting bigger and bigger is really the one thing that will light up a lot of your dorsal visual pathway and initiate interesting behaviors. So these are different kinds of optic flow. Now what I'm going to show you is the first bit of data from my lab, where we recorded from individual MST neurons in a monkey who is kind of just staring at the screen. There's no behavior here. And we want to know, what is it that makes an MST neuron fire? What sort of stimulus is necessary? And because these neurons are really complicated, I'm going to show you a lot of different kinds of data, and I'm going to try and make it as simple as possible. But if you get lost or if you have questions, please interrupt. So this is kind of a standard tuning curve that you've seen before. This tells you that if I move a stimulus in different directions, I get different responses, and the best response is when the stimulus moves down. And this is translation motion, nothing fancy. Stimulus moves down, the neuron fires. Now, to facilitate the processing of lots of data, I'm going to show you this as a color-coded map. This is the same tuning curve now in color, so red means lots of spikes, blue means none. So again, the neuron responds most when the stimulus moves down. So the next question you can ask is, what happens if you move the stimulus around in the visual field? In other words, where is the receptive field of this neuron? So for translation, this is no big surprise. You have a very large receptive field in MST. This is not like V1 where things are tiny, tiny. You'll find that you get the same preference for downward motion almost anywhere in the lower visual field within a range of about 30 degrees by 15 degrees. This is quite typical of area MST. Okay. So this is, aside from the large receptive field, not very different from what you would find in V1 or MT. The cell is direction selective, and it has a certain circumscribed receptive field. Now, the important thing, the most important thing for understanding MST is what I call spirals. These are the combinations of patterns that are either moving towards you and hence expanding, or are rotating around like in the original MST slide that I showed you. And you can define a space of spirals that's analogous to this tuning curve, where as we go around this circle, the rightward point here would be pure expansion, a stimulus moving towards you and getting bigger and bigger. And then the polar opposite of that on the left side would be contraction, as if it's moving farther away. Upwards would be counterclockwise rotation. Downwards here would be clockwise rotation. And as we go around, we explore everything in between. So something that's moving towards you, while it's rotating, will look like a spiral moving towards you, or a sort of swirling spiral pattern. Right? Is that clear? So we basically tested all these neurons with all these different spiral stimuli in all these different positions of the visual field. And this is one neuron. This is still all the responses of one neuron. And what this neuron does is it tells you when something is moving towards you, because it has a response to expansion that is essentially invariant with position in the visual field. So anytime something is approaching you rapidly, this neuron will start firing. Obviously a useful thing to have. And there are quite a few neurons like this in area MST, as I'll show you. We also tested a third class of stimuli, which I won't describe too much. These are called deformation. We can, again, define a space of possible stimuli and record the responses. The reason we include all three is purely mathematical. The three different types of stimuli form a basis for all linear optic flow. So in other words, you can make any optic flow stimulus you can imagine out of linear combinations of translation, spirals, and deformation. In other words, this is every possible stimulus within the space of motion. So, our goal as experimentalists and as theorists now is to produce a model that takes as input the stimulus that we showed the monkey and produces this as an output. This is actually a very hard thing to do. A lot of people have tried to do this with varying success. So I'm going to tell you in some technical detail how we do this. And I'll first say that these stimuli here, the responses to these stimuli, there's a total of about 200 and something, 216 stimuli there. That seems like a lot of stimuli, but it's actually nowhere near enough to solve the kind of modeling problem that we're trying to solve here, and I'll show you why in a second. But in order to solve this problem, we needed to test many, many, many different kinds of stimuli with each neuron, and to that end, we developed a new stimulus, which I'll attempt to show you, which I will not succeed in, but I have a backup plan, and that is YouTube. For some reason, my student decided to post this stimulus on YouTube. Why anyone would want to look at this, I don't know. I mean, maybe it's something you can look at while you're smoking marijuana or something. You can see it's had 99 views. That's pretty good. There you go. Well, some of us anyway. Okay, so here's the stimulus. The goal is to explore the space of all possible motion stimuli as quickly as possible. So YouTube will show this to us now, and if you watch the stimulus, what you'll see is, okay. So what you should see if you look carefully is every once in a while it'll be an expansion stimulus, sometimes it'll rotate, sometimes it'll have translation. And the point is it's going through these stimuli very, very, very quickly. The idea being you can now explore a huge space of possible stimuli, which you couldn't do in the conventional approach to vision. Okay? Thank you, YouTube. So now, having done that, we have an enormous range of stimuli that we've tested. We have the output of each neuron that we've recorded all this time that the animal is looking at this stimulus. And now we're really in a good position to answer the question, what model takes this as input and produces the spike train as output? Very simple. So the first model that we tried is a very, very standard kind of a model. It's one that in its form can't possibly be wrong. It could be incomplete. I mean, it's certainly incomplete, but it can't possibly be wrong. And that is basically that the cell you're recording from gets input from the stimulus, and, well, it receives input indirectly from the stimulus via some subunits, which we construe to be MT, because we know that MT provides input to MST. So rather than trying to figure out the relationship between the stimulus itself and the MST output, we ask, what is the relationship between an estimate of the MT output and the MST output? Right? Because, of course, MST has no access to the visual stimulus. It only knows what MT tells it, so it's going to try and do its computations on these outputs. Now, of course, we're not going to record from every neuron in MT in addition to the one MST neuron we're interested in. We're going to develop a model of how MT should respond to this stimulus, and as luck would have it, MT is probably the most thoroughly studied chunk of cortex in the history of the universe, so we essentially know an awful lot about MT, and we have very good basis for constraining such a model. So our MST cell, we imagine, has the choice of any MT inputs that it wants. It sums them up, and it puts them through a simple nonlinearity to produce the firing rate. The nonlinearity just basically says the output can't be a negative number because we're talking about spike rates, and that once you increase the input, the output goes up accordingly. Here are some more kind of hairy details, which I won't get into, but if anybody has a real interest in this topic, I encourage you to discuss it with me or maybe even join my lab, because this is essentially a very, very hard optimization problem that we bring in a lot of tools from machine learning to try to solve, and I think we've done a pretty good job, but I think there's always room for improvement there. So rather than go through all that, I'll just draw this as kind of a picture. So let's imagine that the MST cell wants to respond to some kind of stimulus, and it has access to all the MT units that exist that have different receptive field sizes, preferred directions, preferred speeds, etc. And it's just going to form connections with them in the way that you would do in a standard kind of neural network approach. The other technical detail that I'll mention is that we're fitting this with cross-validation, which is very, very important. Cross-validation means that you fit the model on a subset of the data and then test it on another subset. The advantage of that is it prevents you from fooling yourself. What this modeling approach will do is it'll just very happily keep adding MT subunits into the model forever and ever because it'll get marginally better and better fits. Cross-validation protects you against that because if you're adding lots and lots of subunits just to fit one chunk of data, it won't successfully predict the responses to the other data, the other inputs, unless it's a really essential part of the model. Okay? So the upshot of this is that we can be confident that the models that we get back are realistic in the sense that they use realistic estimates of the MT output and that they're kind of parsimonious, that they're using the minimal number of subunits that they need to fit the data. Of course, what this really is a giant Hubel-Weasel kind of expedition into essentially taking the very intuitive idea that you can record from a neuron that has certain output properties which aren't present in the input, but you know the properties of the neurons that provide input to the neuron you're recording from. And the question is just how do you arrange all these inputs to get your output? Just like Hubel and Weasel did intuitively in V1. Okay, so the upshot is we've got now our new super fancy stimulus. I got lots of really smart mathematicians in the lab to try and fit these models and solve this problem. And the result is that it doesn't work at all. So here's the data. And as I said, our goal would be to produce a model that essentially perfectly reproduces this data. And here's the output of the model after all the training and everything we could possibly think of to try. And you see that it reproduces the responses to translation. It is, in fact, tuned for downwards motion over most of the lower part of the visual field. But that's about it. It doesn't reproduce really the strength or the pattern of responses to spirals very well. It's hopeless with this deformation stimulus. So that's the end of the talk. Thanks. No, I'm just kidding. So in a way, not to push this too far, but this is actually kind of like what we saw in MT when we anesthetized the animal. You have essentially a very dumb response pattern which represents the very local responses that you saw from the lower level areas. They're sensitive to low level stimulus features, but this model has no intelligent way of integrating them. So, on the other hand, as you now know, it's kind of a dumb model. I mean, it's not wrong. It's true that neurons integrate their inputs, but it's very incomplete. And as you've heard from several speakers here, the one thing we do know about neurons is that they talk to each other even within one area. There's extensive interconnections, and these conversations among the neurons actually do something. So our kind of zeroth order model of an MST cell is that it's getting input from lots of different MT cells that have certain preferred directions, but that each one is kind of an island unto itself. It just filters the input, produces an output, MST sums it up, and that's it. But what if these neurons are doing something more, as we know that they are? What if there are all these properties that have been found by experimentalists over the decades are important, things like center surround organization, center surround suppression, contrast gain control, normalization, synaptic depression. All of these things are known to exist, but they're not factored into our MT model at all at the moment. The MT cells are just simply standalone units. And so, for instance, the one that everyone seems to like and the one that I actually preferred going into the project was, imagine that an MT cell simply gets inhibitory input from its neighbors, which we know that is very common in MT. And intuitively, you'd say this is exactly what you need to produce the kind of selectivity we see in MST, because what does this organization do? It suppresses the responses to pure translation, which I showed you on the previous slide were overemphasized in the output of the model that we have. So rather than building just a model of surround suppression, we realized from reading the modeling literature that actually all these influences, all these kinds of things like synaptic depression, normalization, surround suppression, mathematically they're all kind of the same thing with different parameters. They're all ways to take a single subunit and have it interact with its neighbors with varying parameters that involve the strength of the interaction, which is one parameter, but also the tuning of it. So in other words, you could have this one subunit getting input from neighbors that have exactly the same tuning as itself, or from very broad tuning, or with no tuning at all. And this would be an example of what's often called untuned normalization. Or you can vary the spatial extent of this input pool, so you could go from a very broad set of inputs, like we see with surround suppression, to something that's very, very local. And I've written here synaptic depression as an example of something like this. And it's not technically a case where the cells are getting inputs from their neighbors. It's just a case where you have the output of a subunit will be much, much smaller than you would predict based on its pure feed-forward sensory properties, for the simple reason that when you really, really drive the subunit, it gets tired of firing, and it fires less than you would predict. Right? But the key here is that this sort of effect would be very, very local and very, very tightly tuned, because it's essentially a mirror of the subunit itself. And you can have anything else in between. This is essentially a two-dimensional space that covers almost all of the models that people have talked about in the literature on nonlinear interactions within an area. So, because we have lots and lots of data, and smart students fitting it all to models, we can just go back and refit all the models and say, take the same approach we had before. Let's let our MST cell get input from any MT cells at once, but now we'll assume that the MT cells have some kind of additional influence, and we'll let the model figure out what it wants it to be to optimize the fit to the data. But it could be anything that it wants. So when we do this, it really helps. I mean, a lot. And the main reason I say that is that you suddenly now have really good tuning for expansion motion. This is still this one cell, which I like quite a bit. It's not perfect if you compare just by eye the fit between the data and the model, but it's much, much better. And it's better in the sense that you now have largely spatially invariant tuning for this kind of elaborate stimulus, while preserving the selectivity for the simpler stimulus that it had before we introduced this additional twist. So now the question is, what did the model come up with? How did it solve this problem? And I said that intuitively you would think this would be the answer, but you would be wrong. This was the answer. So when we went back and looked at the parameters that it chose, it always came back, no matter how we ran the model, no matter how we optimized it, it always came back to saying that the very best thing you can do to fit the data is to have a source of suppression of the output of each subunit that is very, very local and very, very tightly tuned to the same thing as the stimulus, which to me kind of screams out synaptic depression, but we have no independent evidence of that. So across the population of the MST neurons, this was almost always the case. If we asked which size of suppressive influence did you want, it almost always came back as that the model wanted it for every cell to be exactly the same size as the subunit itself. And what sort of tuning did you want? It says we want it to be very tuned, and we want it to be tuned for the same thing as the excitatory subunit, with the exception of another mode that popped up here, which we can talk about, where there was a bunch of cells that actually preferred completely untuned kind of modulatory influences. But I think these were less interesting. They tended not to be tuned for the more exotic kinds of stimuli that we're interested in explaining, so I won't go into detail about those. But the upshot is that almost every neuron that we recorded from, we could get a very good model for it if we used the standard feed-forward approach in combination with this one additional twist, which is a very local and tightly tuned modulatory influence. So this kind of brings it back to Hubel and Wiesel again. Hubel and Wiesel had a very simple model of simple cells, and they had a simple model of complex cells. The complex cell was one that basically, again, just added up its inputs. But in order to make it work, they had to first filter it through a nonlinearity. The nonlinearity basically says, don't just add these two up because they'll kind of cancel out. If you're familiar with the Hubel and Wiesel story, you know this. You need a nonlinearity that prevents the output of either one of these from becoming negative, which is commonsensical anyway. If they're real cells, you can't have a negative output. So it both makes it more kind of biologically realistic, and it makes it work conceptually. So we thought maybe we can reduce this model, this space of possible model interactions, even more, just down to one parameter in the way that Hubel and Wiesel did. I mean, conceptually, they didn't do this mathematically. And say, well, maybe each subunit can be passed through a one-parameter nonlinearity. The reason we think this will work is recall that the thing that you have to have your inhibitory influence act on is exactly the same thing that the excitatory influence acts on. So essentially, it's just changing the final output of each subunit, and that would be consistent with this compressive nonlinearity. People very often use expansive nonlinearities in their models. These are the popular energy models. And of course, a linear model is something that could conceivably work, but who knows? So now our MST model is essentially a Hubel-Wiesel model in which each subunit feeds into an integrator, which produces the output of the cell. And each subunit is allowed to have a nonlinearity associated with it. It could also be linear, but with the one parameter, you can choose also compressive and expansive nonlinearities. And the answer is, I guess not entirely surprising based on the previous results, is that essentially all the time, the best fitting model had a very compressive nonlinearity. So a saturating nonlinearity for every cell. And the upshot is that when you add this in, your ability to account for the data jumps up quite significantly in almost every cell. So the cell without any, the model without any nonlinearity does kind of okay. But in just about every cell, we improve the fit of the data by adding this one parameter. And this isn't a trivial consequence of the parameter itself. Because of the cross-validation thing I mentioned, most parameters that you add don't improve the fit at all. There's nothing about just adding parameters that gives you a better model. This is a cross-validated fit, which means that it only gives you a better fit if it actually is structurally important for the model to work. So having done that, we now have a model that we think at least qualitatively captures the main features of the data. It's selective for the right kind of stimulus, and it's invariant with respect to changes in that stimulus. So that allows us to peer a little bit more deeply into how the model organizes itself, and you can see this example neuron that I've been showing you internally has this structure. These are the MT subunits that we find are the most important for driving the output. And so all the complexity that you see here, all these different responses, can really be reduced to two main drivers from MT to MST that sort of one points downward and to the left and one points downward to the right. And those are shown here as the arrows in red. There are other subunits that have less weight, as shown by the translucency of the picture. But it's really those two that do most of the work. Knowing that tells you a lot about what the cell is doing, but it's not enough. You have to know that they're being passed through this compressive nonlinearity. And what that does is essentially say that no matter how strongly I drive this one input, it's not going to drive the output of the neuron by itself. And same thing for this one. You need the two of them together. You need to essentially multiply them. Mathematically, you can think of this like a log, like a logarithm, right? So it's like the sum of two logs is the product. So this is just a sort of sneaky way for getting conjunction detection, the ability to respond only when you get multiple features that you're interested in at the same time, without having to wire up an actual dictionary of all possible feature conjunctions that the cell could be interested in, which would be very, very complicated. So just for fun, here are some other MST receptive fields. Some are very simple. They look like MT cells. Some are quite interesting to look at. They have lots and lots of excitatory and inhibitory subunits. The point of the slide is that if you were to study these cells in the conventional way, you would just say they're all expansion detectors, which is in a way true, but by itself tells you nothing about how the cell works, what it computes, because you can see that the subunit structure is completely different from one to the next. So you really need this much richer stimulus set to understand how the cell transforms its inputs to its outputs. So that's kind of the summary of how we managed to get what I think is a pretty decent understanding of area MST to the point where we can at least capture the So in the remaining time, I'll just mention we published this paper this year. There's apparently a newspaper called the Toronto Sun, which I didn't know about. I don't know if it's a great newspaper. I think it's probably not. But they were nice enough to write an article about this work. I think they kind of missed the point. But anyway, it was nice. I don't know this paper at all. One of the features of the Toronto Sun is that they post their articles online, and then the readers of the Toronto Sun can write in, and they can write comments about the article. So this is an article about our article. So they were writing comments about our article, essentially. And so I was interested to see what the general public thinks of MST, optic flow selectivity. And so this is the first comment that was posted after the study. You can see it's right down here. So I don't know, if English isn't your first language, this means sort of like, I find your result trivial. No duh is a good expression. Okay, so how much time do I have left? Okay, good. I'm almost done. Okay, so anyway, that kind of made me a little insecure because I thought, I don't know, maybe wake up sheeple 1111 has a point. We're still only accounting for like 50% of the data. I mean, there's so much left that we still don't really understand about these neurons. And this mechanism, it boils down to something so simple that it might actually even be trivial. Like, is it really useful, or is it just kind of obvious in a way? But then I thought, no, wake up sheeple 1111 is just way off. Because, the fact is nobody does much better than this. If you look in low-level visual areas, V1, V2, MT, they're accounting for 40, 30, sometimes 50% of the data. So we're way, way farther along the pathway, and we're doing just about as well. And this is kind of an important mechanism. It leads to very specific experimental predictions. And it leads me to the really second big question I want to address is, does this generalize to the rest of the extrastriate cortex? And I know I only have a few minutes to talk about the entire rest of the extrastriate cortex, but I'll show you that the answer is yes. So we went back and repeated the experiment in MT. Now we assume that MST getting its input from MT is just like MT getting its input from V1. Same stimulus, same model, recorded a bunch of neurons. Now we assume the only difference is that the input is from V1 motion energy units. Fit the model and ask, what sort of nonlinearity do you need to get this model to work? And the answer is, again, you always need a compressive nonlinearity in MT. So that's good. V1 to MT is doing the same thing that MT to MST is doing. So you repeat this operation all through the pathway. Okay, here's a really tough one. How about V4? This has nothing to do with motion whatsoever. It's an area that's involved in shape recognition. So we had to change the stimulus a bit. We use these kind of synthetic, kind of geometric shapes, but we stimulate the neurons in the same way, run the model in the same way, ask about the nonlinearity in the same way. We assume that the inputs now are V1 or V2 oriented subunits, and we fit the model and get the answer. What's the answer? Again, you always need a compressive nonlinearity to make it work. Okay. Last question is, what about V1? V1 is the source of all this input. So let's go do the experiment again in V1. Now the stimulus is little spots because we have tiny, tiny receptive fields. These are V1 complex cells. So we assume, based on Hubel-Wieselology, that the inputs are just basically simple cells into complex cells. Ask what subunits do you need as simple cells to fit the data, and what nonlinearity. You don't get the same answer. So the answer here is that a linear integration works just fine as long as you have a threshold in exactly the way that Hubel and Wiesel envisioned. So with that, I will say that the standard kind of feed-forward model put forward by Hubel and Wiesel and used by everyone in the world since in various mathematical forms works just fine for V1. And here's a mathematical expression of it. It doesn't work very well for the extrastriate cortex unless you add in one little additional twist, which is an input nonlinearity, which, as far as we can tell, is essentially always a compressive nonlinearity. And I'll skip this and get to the conclusion. The upshot is that the extrastriate cortex is important for conscious perception, to try to bring it back to consciousness. You can't really perceive faces, complex motion patterns without it, and you certainly would lose a lot of the important aspects of perception, like the invariance to different conditions, without it. But the models are very simple. You can use what are essentially old-fashioned feed-forward models, and they work just fine. By just fine, I mean they account for 50% of the variance, so there's certainly plenty of work to do. It could be that these issues of synchrony or long-range connections matter a lot. The nice thing about our approach is that if anyone out there has a model they think is better, you can send it to us. We'll run our data through it and give you back the answer. If it becomes 80%, then you win. But we've tried many different frameworks, and so far nothing can beat the really very simple model that I've described to you. And that's the compressive nonlinearity. If you poke around in models of the visual system, you find a compressive nonlinearity all the time. So I'm not claiming to have discovered it, but often it's buried with a lot of other stuff, and it doesn't get the emphasis that I think it requires, because in our case it appears to be both necessary and sufficient by itself to account for many of the properties that intrigue us about the extrastriate cortex. And in fact, it turns up in the original form that I came across it, and in the most thoroughly studied form, it turns up in the locust and in flies and beetles. And so, I mean, if this is a talk about consciousness, I'm essentially telling you that the important features that maybe feed into consciousness to provide you with the visual experience of knowing what's out there are found in almost the identical form in insects, which is possibly an interesting point for discussion. Certainly would tell you something about the evolution of consciousness if it were really true in the way that I'm imagining. And maybe that'll come up later. Yes, this is the last point that I made. So I'll just mention that most of this work was done by Patrick Minot, who is also unfortunately graduating, looking for a postdoc. It's part of a larger collaboration with Dan Butts, who's a really great computational guy in Maryland. The recordings for MST, V4, and MT were done by these guys, and there's the funding. Thanks.